Hi team, chapter 15, the special senses, the eye. This is the third video for the eye, third and final video for the eye. So I want to review before we get into the eye. I talked about this topic, neurons and their processes, when we started the nervous system. So this is my neuron of interest. So here, these black things, are the dendrites. Here's the axon. Here's the axon terminals. And then these green things are other neurons synapsing with the major neuron. So these lines, I didn't want to draw the dendrites in detail, so I just put these little lines out. These are the dendrites. Here's the axon, of this neuron, the green neuron. Here's the axon terminals. And when we talked about this initially, when we started talking about the nervous system, we said that, for example, this neuron, this green neuron, will send an action potential to its axon, down its axon to its terminals, and then through a bunch of biochemistry and electrophysiology, the neurotransmitter is released. Neurotransmitter binds to a channel, and in this case, it's a cation channel, so positive charges flow in. So that would be probably positively charged sodium. So sodium flows in, and at the dendrites and the cell body, as the positive charges flow in, now come over here to this graph, the resting membrane potential, which is here, starts to become depolarized, or we could say less negative or more positive, as those positive sodium ions are flowing into the cell. So you get this depolarizing graded potential. And you can see here the depolarizing graded potential only goes to about minus 60 millivolts, in contrast to the classic action potential that travels along the axon that can actually depolarize to about plus 30 millivolts. So this would be a cartoon of an action potential. And that would happen in the axon. So just remember that the action potentials can only happen in the axon because that's where the voltage gated sodium and potassium channels are. The graded potentials can only, or they're also called local potentials, they can only happen in the dendrites and cell body. That's where graded potentials occur. Also, if this green neuron down here were to release a neurotransmitter and opened a channel that let negative chloride in, then that means your graded potential would get more negative. So here's your resting membrane potential in this neuron cell body. Negative chlorine comes in and you get this hyperpolarizing graded potential. Okay, so now with that in mind, we'll go to the rods and cones. So here's our cartoon of light coming in to the eye, travels through all those barriers to get back here to the retina, and then they enlarge the cartoon. So this layer here, this is all retina. And then these yellow things are the axons of the ganglion cells that will travel in the optic nerve. And so if you look at the retina up close, it's the retina borders with this choroid. So remember that, well, let me go back to this cartoon. This layer right here is the choroid layer. It's deep, the choroid layer is deep to the sclera, and then deep to the choroid layer is the retina. So, and then with the retina, there's a neural layer, and then there's a pigmented layer. So here's a pigment cell. This whole thing is a pigment cell. Here's the nucleus of the pigment cell. And then you can see that the neural layer of the retina includes the phototransducing cells, or you can also call them the photoreceptor cells. So the purple things are the rods or rod cells, and then here's the cones. And you can see with both rods and cones, you have an outer segment that's buried in the pigment cells or the pigmented layer of the retina, and then you have an inner segment. Here's a rod cell body, here's a cone cell body, and then here's the connection with a synapse with these bipolar cells. So basically what we're going to talk about is the light waves come in, and you're going to get so the light waves will come in here and they'll perform all their biochemistry out here in the outer segment of the rod and cone cells. So rods and cones are just modified neurons. So they're going to act a lot like the neurons I talked about at the beginning of this video. The outer segment contains visual pigments. So their visual pigments are molecules that will change shape when they absorb light. And then rods and cones connect to bipolar neurons, or they synapse here with bipolar neurons. So here's our cartoon. The light comes in, goes through all the barriers, gets back here to the retina. And then it shows here in this cartoon, the light is coming in this way, because remember the light rays are trying to get here to this outer segment so they can do their biochemistry and let these cells know that light waves have arrived. So these discs, they're like stacks of nickels, kind of. That's why I like to think of them, because nickel's one of my favorite coins. So they're like stacks or discs, and within the discs are these visual pigments. And we'll talk about biochemistry in a second of the visual pigments. So here's our cartoon. Here's the pigmented layer of the retina. So it contains all these pigment cells, and these purple things are pigment cells, pigment cell nuclei, and then these brown things are pigment. Here's our photoreceptor layer of cells, including the rods, purple rods, and then the yellow cones. And then you get the rods and cones are synapsing here with the bipolar cells. And then the bipolar cells check it, they synapse with the ganglion cells. And then the ganglion cells' axons will send action potentials through the optic nerve to get to the brain. Rod cells or rod photoreceptors are very sensitive to dim light. That's what you use. You use your rods to see in dim light. You see in shades of gray. Also, you use the rods for your peripheral vision. Your peripheral vision is actually in shades of gray. The pathways converge. The rod pathways converge, so the resulting is kind of a fuzzy image or an indistinct image that you see in dim light. Okay. The cones, the photoreceptors called the cones, need bright light to be activated. So here's a cone. They have really low sensitivity, so they need really bright light to stimulate them. They have one of three pigments that gives you a colored view of the world. And they have non-converging pathways. They do a one-to-one synapse. So here is a cone. Well, where's a better one? This is a better cartoon. Here's a cone doing a one-to-one synapse here with a bipolar cell. Okay, so we went through these graphs at the beginning of this video. So if you have a depolarizing, you can have a depolarizing graded potential, or you can have a hyperpolarizing graded potential. Depends on what channels are open. Okay, so we've got our cells labeled. We've talked about the rods and cones, the bipolar cells, and the ganglion cells. But there's also these cells called amacrine cells and horizontal cells, and they kind of they help modulate the information, the visual information. So the photoreceptors, the rods and cones, and the bipolar cells only generate graded potentials. They don't generate action potentials. The only cells that generate action potentials are these ganglion cells. And the ganglion cells send their action potentials down their axons to give you information about vision. And I tell you here, ganglion cells generate action potentials that are transmitted in the optic nerve. Here's a table comparing rods and cones. I like tables to help sort of bring all the information together. So now we're going to talk about this amazing process called phototransduction. So all that means is you're taking photons of light and you're transducing that physical information, the photons traveling in waves, into electrical information. And here's our heroes that do that. So the light comes in, light waves travel all the way back here, like I said, to the outer segments of rods and cones. This is where the biochemistry is going to be happening. And then you'll get a graded potential in the rods and cones. They'll synapse with these bipolar cells. They'll do a graded potential. Bipolar cells synapse with ganglion cells. Ganglion cells do action potentials. So now here, remember I told you these outer segments are made up of these discs, kind of like stacks of nickels. And then if you cut out a piece of that disc, you can see, here's the plasma membrane. So here's a phospholipid bilayer of the outer segment of that cell, because all cells have phospholipid bilayers. This is just a distinct motif. The phospholipid bilayer is arranged as a series of discs. But anyway, within the phospholipid bilayer, you have this molecule called rhodopsin. So this big purple molecule is called rhodopsin. And it consists of, well, it's a visual pigment, and it consists of one part called retinal and another part called opsin. So rhodopsin, retinal plus opsin equal rhodopsin. Retinal itself is a light absorbing molecule. So this molecule in here, this is retinal in here in the cartoon. It absorbs light. You can make it from vitamin A. And where do you get vitamin A from? Carrots. And what do you want to eat when you want to see better? Carrots, because it has vitamin A. Vitamin A is used to make this molecule called retinal. And retinal is a molecule that will change its shape in the presence of light and also in the absence of light. So it's a light detecting molecule. Okay. So remember we said here's our hero molecule. Here's rhodopsin. Remember it's embedded in the outer segment plasma membrane of the rods and cones. This is just showing you the biochemistry. So in step 1, it says pigment synthesis. It says 11-cis retinal. So this is the shape. See, check out the shape of 11-cis retinal. It's bent. It's derived from vitamin A, and it combines with this purple molecule opsin to form rhodopsin. So, and then this shows you too, this is what rhodopsin looks like in the dark. So in the dark, retinal is bent. Step 2, it says pigment bleaching, so that the pigment is rhodopsin. So in the light, so you're over here, the light rays come in, retinal absorbs the light ray, and rhodopsin then goes through a series of changes in which retinal, 11-cis retinal, the bent retinal, when it gets a light ray, will straighten out to a molecule called all-trans retinal. So see up here how it's bent, retinal is bent. Now in the presence of light, retinal is straight. And then when retinal is straight, it can release itself from the big purple thing here, opsin. Now if people that have malnutrition-induced blindness, you can take vitamin supplements. So vitamin A supplements can actually reverse the degenerative changes to malnutrition-induced night blindness. So just keep eating your carrots and a vitamin A rich food. Now, in the dark, the pigment will regenerate. That means there'll be enzymes there to convert the straight or trans retinal back to the bent or cis retinal. And then more light comes in and you can do the whole process again. So, like I said, the visual pigment of rods is rhodopsin. And then the visual pigment of cones is photopsin. Cones works pretty much, this biochemistry that I just talked about works pretty much the same way with cones. So we're just going to talk about rods. In the dark, a rhodopsin forms a cis retinal or bent retinal and accumulates, and then the rods get activated in the dark. Remember, rods are activated, or use rods to see in the dark or dim light, and use cones to see in bright light. Then when light is absorbed, so the light rays come in, light gets absorbed by 11 cis retinal, then what happens is when 11 cis retinal straightens out to all trans retinal, it can break down rhodopsin. That means retinal and opsin can separate. That's called bleaching of the pigment. So at that point, the sensitivity of retinal is diminished, but it's in the light, so you gain visual acuity. Dark adaptation occurs from moving from bright light into darkness. So you've noticed that if you go from bright light into darkness, your pupils are going to get bigger. So when you're in the dark, the rhodopsin builds up in the rods when you're in the dark. And then the rods will kick in to help you see in dim light as you get more and more rhodopsin. So if you go like from a really bright room to a really dark room, you don't see well. Well, it doesn't have to be really dark. So you go from a bright room into a dimly lit room, you don't see well, but then over the next couple minutes, you start to see more clearly in the dim light. Obviously, if it's pitch black, you can't see anything, but if it's dim light, your rods will kick in. Your cones, if it's low level light, your cones don't work. Okay. When you move from the dark into the bright light, your pupils constrict, obviously. Large amounts, when you get into the light here, that's this yellow part over here, large amounts of pigment are broke down instantaneously. So this rhodopsin breaks down really quickly in the bright light, and then your rods cease to function. But your cones rapidly adapt, and within the next 5 to 10 minutes, you start seeing better when you've moved from darkness into bright light. So this is a cartoon from your book if you're using Marieb. So here's the outer segment of a rod. Take a little piece out of that and enlarge it to this panel of cartoon. And then it gives you step by step. So number 1, retinal. So here's retinal. See, it's bent inside opsin. So retinal and opsin together is called rhodopsin. Here comes in the light wave, and that energy of the light is absorbed by cis retinal, and then cis retinal straightens out. It changes shape to all trans retinal. When that happens, this G protein called transducin can bind to this area, and then it can become activated. So the visual pigment activates this G protein called transducin. Transducin then shuttles along the plasma membrane and activates this molecule called a phosphodiesterase. And a phosphodiesterase, what it does, it takes cyclic GMP into the molecule called GMP. It's like ATP, only it's GMP. So PDE converts cyclic GMP into GMP, and that causes the cyclic GMP levels to decrease. So when rhodopsin absorbs light, what you're doing is you're decreasing your levels of cyclic GMP. And then in the dark, the cyclic GMP gated cation channels are open, so sodium can come in. You get a partial depolarization. But when light's there, you decrease the amount of cyclic GMP, and then you close the cyclic GMP gated channel in the light. This is just another way to, this is from another book, to show you how phototransduction works in the dark and the light. This is from another book, whichever one you like the best. So basically, excitation of cones. So remember, these guys are the cones. Cones are active in bright light, and they become inactive in dim light. There's 3 types of cones: blue, green, and red. You can activate more than one type of cone at the same time, and you get these intermediate hues or colors. And if you have a congenital lack of one of the cone types, then you're colorblind. Okay, now we're going to talk about the visual pathways. So this would be like a transverse section through your head, and then we're looking superiorly down on that cut section. So this, here's your right eye, here's your left eye. This just shows the visual pathway. The left eye can only see this much of an area, and then both eyes can see from here to here. And then it shows the optic nerve. So this is the optic nerve. So it's got the axons of those ganglion cells running in it. And this whole area is called the optic chiasm. So this is the optic chiasm. So that what happens in the optic chiasm is some of the axons of the optic nerve cross over. So for example, if you look at the right eye, follow the blue neurons, they actually cross over, so they decussate, or the optic nerve hemidecussates, because half of the fibers or axons cross over in the optic chiasm. But look, half of the axons or fibers from the right eye do not cross over in the optic chiasm. And it's the same for the left eye. And then you have these nuclei, so the suprachiasmatic nucleus, the pretectal nucleus, the lateral geniculate nucleus of the thalamus, the superior colliculus, all these places where you can synapse on your way to get information back here to the occipital lobe, or the primary visual cortex. Okay. So axons of ganglion cells form the optic nerve. That's what I said on the last slide. The medial fibers, so this would be medial, the medial fibers of the optic nerve cross over. So here you have crossing over the medial fibers. Here you have crossing over the medial fibers from the left eye. So they decussate. The whole process is hemidecussation because you only get half of them. But those fibers decussate at the optic chiasm. The lateral fibers, so these guys out here, the lateral fibers do not decussate. They do not cross over. The medial fibers of one eye here, so the medial fibers of one eye join the lateral fibers of the other eye to follow the same optic tract. Most fibers of the optic tract, they go to the thalamus. Remember, the thalamus, that's the gateway to the cortex. If you want to get into the cerebral cortex, you want to get into the visual cortex, you have to go through the thalamus. The optic fibers from the thalamus connect. So then, let's see, where's our? in our cartoon. Well, the optic fibers from the thalamus, so where you synapse the optic nerve fibers synapse in the thalamus, and then those fibers will go back or go to the rear to the occipital lobe, and specifically the primary visual cortex. Other optic tracts will send branches to the superior colliculus. So here's a superior colliculus, here's a superior colliculus. Because this is a surface view, the inferior colliculus are just below, so you can't see them in the cartoon. And that will initiate reflexes based on what your eyes saw. And then this is a small subset of ganglion cells will project to this pretectal nucleus in the midbrain, and what happens there in the pretectal nuclei in the midbrain is you synchronize your biorhythms with the natural light and dark cycle. So this is involved, the pretectal nucleus is involved with your pupillary reflexes in response to light and dark. And then from there, they go to the hypothalamus. And the hypothalamus basically is your timer for your daily biorhythms. So when is it time to wake up? When is it time to go to sleep? Time to take a nap. And then this bullet says both eyes view the same image from slightly different angles. So if the fixation point is here, this is what you're looking at. The right eye will view it at a slightly different angle from the left eye. And then when your occipital lobe, or where your primary visual cortex lives, when the primary visual cortex fuses the different images that get to it, that's when you get your 3D or 3D depth perception. Okay, so back here, the primary visual cortex, it's going to process and contrast information, object orientation, what you're looking at. And then the visual association area, which is right next to the primary visual cortex, will process the form, the color, the motion input from your primary visual cortex. And then you'll get really complex visual processing, goes into other regions like your temporal lobe. So it'd be over here to help you identify the objects specifically. You can go into the parietal cortex, postcentral gyrus, to process the spatial location of the object that you're looking at. Optic radiations, so these blue guys right here, optic radiations project back to the primary visual cortex, which is where you want to send the information ultimately. But you have to synapse in these nuclei. Conscious perception of vision of what you see reflects activity in this the occipital lobe, specifically the primary visual cortex. And then this is a cartoon, it's side by side with a photograph of the human brain to show the structures.